Got fruit flies with a wild type phenotype having gray and red. So capital G is for gray and capital R is for red. Certain mutations can cause changes to these traits. Mutant flies may have black body and cinnabar eyes. So gray body, red eye, or you might have black body and you might have cinnabar eye. A researcher records the true breeding. Do you know what true breeding means? Do you know what true breeding means?
Like when it's fully.
What?
Like it's only one of them.
It's homozygous. Homozygous for gray and red. So it's going to be capital G, capital G, capital R, with true breeding, black body, small g, small c. And of course, all the F1 displayed a wild type, right? So this becomes capital G, capital R. This becomes small g. This is small r. I don't know why. Right. Okay. Small r. And they all become capital G, capital R, small r. That's the P generation, that's the F1 generation. Now, if you take the F1 generation and cross it with a true breeding black bodies and cinnabar eyes, that means you will take the capital G, capital R, small r with black body and cinnabar. Now you're going to get capital G, capital R, capital G, small g, capital R, small g, small r, small g, small r. And you will get capital G, capital R, small r, capital G, small r, small r, small g, capital R, small r, small g, small r, small r. So now, we're going to take our things and we're going to describe them with a phenotype. That's going to be green body, red eye. Green, is it green or gray? Was gray. Okay. Gray body, cinnabar eye. Okay. And small g means black body. Red eye. And we're going to also have black body, cinnabar eye. So we're expecting 1 to 1 to 1 to 1. That's the expected, right? Now, we're expecting 244 of each. Expecting, predicting is the same thing. We're going to be observing something different. Look, whenever you see these huge numbers, like you see 2 huge numbers and 2 very tiny numbers, this is going to be a prime example of linkage. Linkage is when the 2 genes are found in the same chromosome. And linkage is responsible for disobeying the law of independent assortment. Okay. Now they're saying explain the difference between the expected and the actual numbers observed. Well, look, the numbers observed, provide us with linkage because we have 2 very huge and 2 that are very small. But to support your data, it's best to do a chi-square test and see if we can reject or support the null hypothesis. So of course, null hypothesis always assumes that things are simple, which is that the results, okay, are due to chance. Okay, so essentially the difference between the predicted and the observed is only because of chance, and there is nothing biological behind it. Now, we can support null hypothesis or we can reject it. You should do a chi-square test. If it's less than the critical value, you support null hypothesis that there's chance, right? And if you reject it, that's your chi-square value bigger than your critical value. And now it's time to do the chi-square test. Okay, so now I'm going to be doing observed minus expected or predicted squared over expected, which is the predicted. I do it for the first row. Then I do it for the second row. Then third row. And then the last row. Let's use the calculator. So I got 653 point, just say point, like, okay. So that's my chi-square value. So now I have 654. The question is, what is my critical value to compare to? So, since I have 4 possible results, which is gray-red, black cinnabar, gray cinnabar, and black-red, okay, my degrees of freedom, which I will refer to as DF, is equal to how many possibilities I have minus 1, which is 3. So, let's see the critical value that corresponds to this 3 degrees of the freedom. So if I have 3 degrees of the freedom and my p-value has to be 0.05 unless told otherwise, my critical value is going to be 7.82. And since my chi-square value, which is 654, is much larger than 7.82, I will simply go ahead and I will reject null hypothesis. I'll reject chance. That means the results that I saw are not because of chance, but are truly because of linkage. Now, let's explain those results. The 2 super high values are caused by linkage. And the 2 very small values are caused by crossing over. Okay? And if someone asked you to find the crossover frequency, it's not that hard. Add the crossovers and divide by everything. Let me know how much you get. That's if we're asked to find the crossover frequency. And I got 9.1%. 9.1% crossover frequency.